ये बंद नहीं करेगा, कुछ नहीं करेगा, वो बाकी देख रहे YouTube पे।
कौन देख रहा है?
बहुत वगैरह देख रहा है।
अरे कौन नहीं देख रहा है?
अरे देख रहे हैं बाबा।
अरे एक बार लॉगिन कर देते ना बे।
पागल है
अरे क्या फर्क पड़ रहा है?
अबे क्या है? हाँ वैसे भी टेंशन नीचे आने वाला है।
हाँ पक्का? अगर नहीं आया देना देखना है कितना है।
मैं खुद ही नहीं कर रहा हूँ।
साइन कर दिया अटेंडेंस
नहीं
मैंने इंग्लिश की अच्छी अच्छा
पता है दो में से नीचे
हाँ इसको
लॉगिन कर देना फिर।
वो बहुत
ये डायरेक्शन का कौन है?
खुद भी बोल रहा
इसकी डायरेक्शन ने बोला सकती है कुछ।
अगर किया भी होगा तो क्या नहीं करा भाई क्यों कर लूं
वे चल रहे यार, हम तेरे लिए रुके हैं भाई, ब्रेक खत्म हो जाएगा फिर।
आदित्य
और बाद में तो उसका काम है वो सब। चल नहीं तो चाचा खाएगा। चाचा खाना है
भाई डॉन ने बोल दिया
टूट
गया।
कर दिया तूने कर दिया वेरी गुड
अरे वो लटका सकते तो चलना यार। क्या
तुम्हें गाना बना देना सिमी सिमी
शांत हो।
सिमिया
आगे भी बोल दे।
नीचे
अभी नहीं
पागल है।
अभी नहीं पसंद है
पेंडुलम
अरे अभी नहीं पसंद
नीचे
सिमिया
नीचे चलो ना। अरे सब भूला नहीं तो जा गए अंदर, तो अंदर क्यों आया वो बता।
और पर है भाई
काकड़ी काकड़ी खाते भाई, काकड़ी
और कलर कैसे है
भाई मैं बोल
भाई
चिल्लर की हूं मैं चिल्लर की
चिल्लर पार्टी का चिल्लर की।
ऐसे 20 रुपए
ओ भाई बोला चिल्लर पार्टी का चिल्लर किंग।
ऐसे 20 रुपए
गलत ने बोला भाई गलत ने बोला तो
मैंने
खराब नहीं किया
जल्दी चले यार, ब्रेक खत्म हो जाएगा, बाकी कुछ नहीं है।
ओह ये 10 साल पुराना नहीं है, ये मेरे पैदा होने के पहले बना है।
ज्यादा
ज्यादा
खत्म हो गया।
पुराना है। वैसे तो क्या 10
नहीं चल भाई, चल चल चल
टाइम कर दिया सिमी
लोगों ने बोला
ऑटोग्राफ चाहिए भाई मेरा, देखो तो टी-शर्ट पे चाहिए क्या किसके?
चल पागल है क्या
अरे ऑटोग्राफ दे और चल जल्दी।
अबे मैं अपना ऑटोग्राफ भी सेव कर पाता हूँ, क्रेजी हूँ।
आगे करेंगे अपना सब इम्पोर्टेंट
सिमी
है
सिमी है।
आर्ट का फेवर हो जाएगा
कोई जस्ट साइन कर देना ना भाई। एक बार
साइन क्यों करना है?
वो हाथ से
घर जाके देखना।
हाँ मेरे को टेस्ट है वो देख।
कौन से टेस्ट के?
इंग्लिश में।
मेरा डेटा क्यों होगा?
हाँ
विदाउट डेटा
तो
इंटरनेट।
मुझे डेटा नहीं है।
तेरे पास फोन नहीं है?
मैं बस नया फोन लाया नहीं, वो बैटरी चार्ज करता
भाई प्लीज कर
हूँ।
जा।
अबे यूटी का है ना, दिखा ना यूटी का है वाईफाई से।
कर जाके देखना।
यूटी का है ना वाईफाई।
ब्रो इतने लोग हैं, तो उनसे नहीं मांग सकता, भरत से, आदित्य से,
अबे
अभिप्राय से, तेरे दोस्त
नहीं ये नहीं दे रहा,
क्यों?
पागल है।
दे देना, क्रोम से कर देगा।
नहीं इसकी अम्मी भी चेक कर दे, कुछ डरता है क्या
एग्जैक्टली मेरे को भी वही प्रॉब्लम। हाँ ऑब्वियसली
चलो बाद में फोन ही देगा लास्ट टाइम दिया था ना
नहीं उससे इसका प्रॉब्लम नहीं होता
मेरे पास
बस।
पासवर्ड बता दो तो अपने हाथ से कर। हाँ बेटा
तो उसका ट्रस्ट देख रहा है, तेरे को पासवर्ड बताने को भी रेडी है।
मैंने
ऑन किया या ऑफ किया? ऑफ।
अच्छा ठीक है पूरा तो मेरे ख्याल से देगा बस
सी मान गया, कन्विंस करना आना चाहिए।
गूगल
भाई कितना इंग्लिश में था क्या तेरा?
नहीं ह्यूमैनिटीज था ऑन सुपरविजन
नहीं टेस्ट मेरा टेस्ट नहीं होता इसलिए पूछ।
इंग्लिश और हिस्ट्री दोनों
दोनों के आ गए मार्क्स?
एक
देखो क्या लगता है नहीं सर वो आदित्य ने नहीं किया अंश ने नहीं किया
भैया मेरा कर दो
अद्वैत तो आपसे ले कर दे अंश
ऑफर डालने के बाद तुम मेरे आ रही हो
पासवर्ड तो मत देख
मैं दिखता भी नहीं है तो मुझे किसको समझ में आने वाला है सी पासवर्ड ऑन है उसमें नहीं ना अबे तेरा कॉम्बिनेशन से हो जाएगा
अरे अरे मैं फ्री हूँ निकलाओ What? Just grow?
फोन पे तो हाथ में था
तो ऐसे लग रहा है फेक कैमरा है लग रहा है
तेरा फोन हाथ में था क्यों ठीक करने गया
नहीं
अपने अपलोड करो अगर क्या हो तो मेरे को एक फोन खराब हो गया मेरे पास एक्स्ट्रा पड़ा
मेरी का भी नहीं
साइन कर लेते ये लेना है
तो बाहर है
1943 क्या देख रहा है
मेरा चैनल देख
अभी और ही रहने दो मुझे टाइम पे
आदित्य और चैनल देख
इंग्लिश
तू क्या किया ने लगे बेस्ट में
मैं
हां
भाई मैं
1.5 दो
मैं से बहुत चलता
दो
सही बता कि मैं टाइम हो गया मतलब
अरे मतलब अच्छा से बाल वगैरह
तो
वो
मस्त लगता
हां ना गोरे गोरे
बाल क्या पर ये स्ट्रांग लगता
क्या हुआ इकडचं काढ तिकडचं काढ इकडे करून टाकतो इकडे काय येणार आला
इकडचा तिकडा
अरे नहीं वो बराबर हो गया
क्या अभी तक इधर
हॉलैंड वगैरह
है वीडियो में बट पासवर्ड नहीं है पासवर्ड ही नहीं है तो कैसे मेरे को नहीं पता हमको वाला
क्रैक कर हो गया
अरे मेरा फिंगरप्रिंट चाहिए रहेगा फेस लॉक
आप खुल चुका था
अरे यार तो खुलेगा ही ना पागल आते के पास करो बैंक बैलेंस भी देख सकते हो
अच्छा है भाई अच्छा है ना आधा मार्क में उसके ऊपर
भाई कौन है यार घटिया सा कौन इंस्ट्रक्टर है यार
पीछे क्या था क्या
लॉग आउट कैसे करते हैं
ओ नो
लॉग आउट
मैं क्या
ये लॉग आउट की है ही नहीं करे
सुनो मेरा को पैसे दो
ये लोग खाएंगे डाल
खाली है यार मुझे जेनेटिक्स नहीं पढ़ना है यार जेनेटिक्स कितना अच्छा चैप्टर है क्यों
फोटोसिंथेसिस हो
जेनेटिक्स हाँ फोटोसिंथेसिस
मैम ने बोला था प्लांट्स एक साथ पढ़ाएगी
नहीं जेनेटिक्स बिना दिमाग वाला चैप्टर पढ़ता है ना प्लीज चुपचाप
क्यों जेनेटिक्स अच्छा है पढ़
हाँ भाई तो दिमाग लगेगा ना तो दिमाग क्या चल रहा है
तो 7 घंटे से
जेनेटिक्स
तो 7 घंटे से
मैं डेंटिस्ट से आई हूँ एनेस्थीसिया डाल के आई हूँ मैं मेरा सर दुख रहा है मेरा ये दुख रहा है मेरा ये हाथ हिल नहीं रहा है जेनेटिक्स में कैसा दिमाग लगता है
तेरा हाथ को नहीं डाल
क्योंकि इधर ना इतना तो अच्छा चाहते हैं क्यों उसको करना चुप रहे हाँ तेरा देख लिया है लगता है केमिस्ट्री का है ना परफॉर्मेंस मेरा नॉट मेरा आ गया केमिस्ट्री का
मेरा भी आ गया मेरा एक भी मार्क नहीं गया
वेरी गुड डैम भाई लेट्स गो
बस वो मेरा क्वेश्चन किसका छूट गया
ओ हाँ अच्छा है
मैंने देखा ही नहीं था वो उसके अलावा मैंने
ओ शिट मेरा एक भी मार्क नहीं गया
उसका इसको मोहम्मद पर आएगा
28 मुझे कुछ भी
बहुत है उसमें
ये हाथ ये
Who did it? आदित्य।
वाला सुनी क्या ना
नहीं,
क्या किया भरत ने?
बैक करवा था ऐसे
मैं नहीं कर रहा है कुछ भी।
भरत यार what you are doing
आदित्य
यार?
आदित्य कर रहा था।
क्या
भरत बात है ये। भरत
सारी बातें।
भरत
क्या कर रहा है
मैंने क्या किया?
हट
Wait on.
भरत इधर आओ।
अरे यार को जाने दो
No, भरत you have to switch.
No one of us heard in this
No, Varun was not. No,
I was
Varun
not
was-
I don't want parents to come here and what's going on. ये क्या kindergarten के बच्चे हो क्या? तुम लोग क्या banging the door and all? Who did that?
I was not banging out I was just coming and then
See whatever it is, let us have a bit of little थोड़े कुछ etiquettes हो ना school class कैसे होता है वैसे.
मुझे अंडरस्टैंडिंग
अब कर रहा था बस मैं
कोई भी करे दोनों के लिए बोल रही हूँ. I don't want this blame
Not me.
game. Okay, okay, not Abhipraya, both of you. And I know who has done. Okay? So better don't do that. And please, I'm repeating, थोड़ा sincere रहो यार. 10th standard है बच्चों लोग यार. What is this now? You see, I am juggling from here. वहाँ जाओ, यहाँ जाओ. अकेले कहाँ टेकूंगी मैं? And are you doing your homework, everybody?
यस
Yes, ma'am.
पक्का. I don't want any parent to come here and tell me कि homework कर नहीं रहा है बच्चा उस पे special attention दो करके. Okay? You have to finish everything. जो class का है वो class का. There is that other.
अंश
Ansh doesn't write. Where is Ansh?
मैं भाग गया हूँ
See, all the teachers are going to update me from henceforth कि कौन बच्चा class में क्या कर रहा है. Okay? So make sure I have to make sure कि मेरे जो batches हैं वो अच्छे हैं. I don't be like my own. I'm going to treat you like my own kid. So accordingly you have to treat me also. ठीक है ना? कि हमसे कोई complaint नहीं आनी है. Let's be the best batch. मस्ती करने के लिए मना नहीं कर रही हूँ, पर ये धाड़-धूड़ क्या है? हाँ? Running around like toddlers. What is all this?
मैं क्या किया?
Abhipraya. Don't do that.
चलो मुझे नहीं कर मैंने कुछ किया
मैं किसी को point out नहीं कर रही हूँ, बस थोड़ा ठीक से रहो ना. And Laksh, why your phone was ringing during the class? From tomorrow onwards, no, please keep deposit your phones. ना मेरे यहाँ पर रख दिया करो. वहाँ पर
हम जहाँ के बचते हैं वो उसमें रख देंगे आपके
हाँ?
जिन लोगों के फोन बीच में बचते हैं उनके आप बोलो बता
अच्छा. तुम लोग को ना बहुत ज़्यादा छूट मिली है. Only if you make 30 From 1st June, no, the phones are going to be there with me. I'm not going to. Exactly.
Done,
किसी का
phone भी
नहीं
था।
लेके मत आओ. But अगर लेके आते हो तो it will be with me.
क्या करता है तुम लोग? उड़ा डालूंगा।
ए ज़्यादा हो रहा है
कैसे डाउन होते थे उनके इसलिए क्या नहीं मैं
नाम पे देखते हैं नाम क्या
अरे
है
यार
बॉयज
बॉयज
अभी
दूसरे
दूसरे
क्लास का वो है
गैंगस्टर है नंबर नहीं सुन लिया
फ्रेंड्स भी लोग हैं
भाई देखो इन सबका नाम
मेरे पास नहीं
अभी वाला भाई भाई
देख मैं इन दोनों को ऐड कर दूंगा यहाँ पे
मुझे ऐड कर
तेरा नंबर
मुझे भी ऐड कर
तो मेरा भी है दूसरे
इसका नंबर है ना सास के वाला लिख के ले
अरे अभी क्या लोग
सास के कुछ अरे मेरा फोन भी हो
घर
गया
जाके
घर जाके लेता हूँ फोन पे लिखता हूँ ना
उसमें ऐसे नहीं गए
इसलिए मतलब टाइप तो कर सकते हैं ना
मुझे नीचे आएंगे ना तो फिर तुमको नंबर दे देना मैं लिख दूँगा और तेरा तो लिखता हूँ सेव कर लूँगा
क्या
लिख दो
तेरा मम्मी का नंबर है ना
नहीं मेरे
तो तेरा अपना नहीं भेज तुम गणेश जी के पीएफके
याद नहीं चल
भी देख ले
वो सिग्मा
अभी देख ले
तो अपने मम्मी का नंबर
जब फोन लाया था तभी मैंने प्रोफाइल फोटो सेव किया
उसके बाद नहीं चेंज करा
और
सेम यार मैंने 7th में किया
2019 मैं तो फोन ही
मेरा
अरे
मेरा
लगा
पार्सल कोड मिला तो उसके बाद मैंने एक बार प्रोफाइल पिक्चर
Same.
उसमें चेंज किया
मैंने भी 7th
I don't even
हाँ, गणेश जी का है।
remember.
ये ना
मैंने तो अपना
2019 दिया 2019 से एक बार मुझे बोल रहा है मैं कैसे नहीं सर कुछ भी नहीं है मेरे फोन में
कुछ भी नहीं है
एब्सोल्युटली मत
आज-
अबे नहीं मत
अरे भाई मैम नहीं मत कर सीसीटीवी
क्या है
अच्छा वो कर रहे हैं क्या
एक मिनट और क्या है
क्या
बलवान
I'm judging you. Regret नहीं मैं judge कर रही हूँ तुझे.
बता अबे वो मिशन क्या नाम
ऐसा कौन सा मिशन है
भाई
हाँ ये नाम है बस सेव फाइल
ओ
इनकॉग्निटो एंट्री होता है
नहीं होता यार नाम होता है
यार स्टार्टर मैंने कितने सुंदर
माइनक्राफ्ट सर्वर है
पता है
ये सब मेरे लाइन का था ठीक है वो स्टार्टिंग के एक दो चैप्टर हाँ उसके बाद
नाइस
गुड आफ्टरनून मैम
Sit.
lots of the time क्या है
Very fast, complete this set of questions and we move on now to the next chapter. So what is left from here? E sub number 1.
बढ़िया
How many more left from here? Let us check. So this you can take as homework, I believe. E sub number 1 pick karo.
right
Let's
now
see
or
some
work
homework. Now check. From the new book, E sub number 1, pick this question. You will try as homework. If there's a doubt, next class you will tell me. E sub number 1, 4, number 5, 6 and 7 done, no?
Ma'am, is there any problem with the history sir checking papers?
History.
Yes, sir.
Your problem.
No, I'm just asking because last time chemistry had same problem, so history is also delayed, so I'm just asking.
Oh, you got less marks.
No, I'm just asking.
अरे मैम को कैसे
Some
बुला
paper,
रहा हो
अरे नहीं मैम ने बोला लास्ट टाइम chemistry sir बीमार है इसलिए वो करेंगे.
उतना ठीक है बताओ ये कैसे बताओ कोई दिक्कत है नहीं
बताओ.
हमको तो
Problem क्या है बोलो then I can pass it
मैं
on.
बोल रहा है कि हिस्ट्री में बताओ आएंगे बस चेक हो गया
मार्क्स for the E first one मेरे पहला
अभी तक नहीं आया.
MCQ है
कौन सा है?
E first one तो हमको
E first one. अच्छा तो let me just finish this. 6 हो गया ना? Number 6 is done, no? 6, 7 done and 8 is left. So 8 also you will do as homework. Now tell me which one you are saying? E sub number 1.
yeah A has grease on both sides B has grease
So
on both
figure
sides
leave A coated with grease both sides, B lower,
C upper
C upper,
D none
D nothing. Which leaf would dry first? Which leaf would dry first?
I think D
To dry out, what is the condition?
so
It should not be covered. Then only
yeah
it will
so
dry out.
D right
So, without application. So, D is answer number 1. Give reason. What reason will you write? The stomata
No, grease
are
was
not
coated.
blocked. Stomata is not blocked. So, there is enough loss of water happening. So, this will dry out first. Because, why dry out? Because you have taken out from the plant. It does not have any more connection with the plant. That's why dry out. Otherwise, if it is connected with the plant, then no dry out.
dries last will be
Dries last, both surfaces. Both surfaces. Dries last, both surfaces. So, what happens?
means copying your time doing
Acha, now, for the bio test, the 2 chapters what we are already done and what your portion is there any doubt?
no
Doubt hai? Did you read the chapters? Will you be able to appear for the test on Sunday?
yes
Did you go through the chapters? Doubt hai, coach?
Ma'am, I have forgotten you had explained कि mitosis में 2 होते हैं और meiosis में 4 होते हैं because meiosis does mitosis and death अलग मैं वो पूरा ही भूल गई हूँ और अब मुझे टेक्स्ट बुक से नहीं समझ में आ रहा
बट वो meiosis का नहीं है वो उतना नहीं है
अच्छा, ठीक है.
है हमने डिस्कस किया था वो
अच्छा, ठीक है. I'll give you a short test. 10 minute का एक test देती हूँ. उसके बाद both the chapters which you are going to write for
sure
test on Sunday. Notebook में लिखो. Textbook बंद करो. Notebook में लिखो. And this one will come here. Here, you move that side. Give him space. And all of you will wear a light jacket.
लाइट जैकेट पहन के आओ, फुल स्लीव टी शर्ट पहन के आओ,
लाइफ जैकेट
लाइट LIGHT स्वेट शर्ट्स पहन के आओ, don't come with a very thin t-shirt and sit here and don't ask us to put off the AC. We are constantly talking, we start feeling hot. Very true I'm speaking. Nobody will ask the teachers to put off the AC. Is that clear? You come prepared and eat a lot so that the body heat is also maintained. Then standard students,
Orange nose bleed.
huh?
Orange nose bleed is seen.
My nose bleeding is because of the heat. For that I cannot tell him anything. That he is not doing on his own. But you make yourself strong. Your blood should boil now. Your blood should not be so cold right now. This is not the age where you should be feeling cold. So then I have to check whether you are 16 or 60. Number one.
59.
You're not 16?
You're 15.
है?
15.
Okay.
I'm 14.
भाई चलो
मैं बर्थडे साल में शुरू
तो जब वो चॉकलेट खिलाएगा उसका मतलब वो तो
हाँ, शुरू हुआ है।
चलो स्टार्ट. Number
मेल हो
one.
गया ऐसे ठीक है
So let me take a short recap on structure of chromosomes. You write very fast because I want to start with the next chapter today. The first question, structure of chromosomes and absorption both I give you. Number one, what are nucleosomes? What are nucleosomes? Write test. Class test you write and put today's date. What are nucleosomes?
well not
Question number, question, put a question mark, continue the same question. What are nucleosomes?
अरे
What is meant by histone
Same
octamer?
question, right?
Same question. What is meant by histone octamer? What is meant by histone octamer? Leave 2 lines, 3 lines for the answer. You will write exactly in this format that I am telling you. Don't write all questions. 3 lines for the answer. Go to question number 2. Go to question number 2. What are the proteins present in the centrosome known as? What are the proteins present in the centrosome known as? Question mark. What is their function? Question mark. What is their function? 3 lines for the answer. Number 3. 3 lines for the answer. Number 3. Question number 3. Draw the stage of mitosis. Draw the stage of mitosis. That suits the following features. Put a dash.
For this animal or
That from the question you will have to understand. Number A, the first feature. First feature. The cell wall, the cell membrane shows furrow. Cell membrane shows furrow. Number B, duplicated chromosomes.
bleed space.
No, just write down A, B, C. A, I gave you one condition. B, duplicated chromosomes separate.
this is C.
Number B, duplicated chromosomes separate at the centromere. Duplicated chromosomes separate at the centromere. C, sister chromatids. Sister chromatids move towards opposite poles. Sister chromatids move towards opposite poles. Last, number D. The cell at the prophase stage has six chromosomes. Cell at the prophase stage has 6 chromosomes. So these 4 conditions you'll have to think of and draw the suitable diagram.
These are 2 marks each.
Huh?
3, 4.
Diagram.
Half, 2 marks.
Total 2 marks.
2 marks. 2 marks for the diagram. 4 lines for the answer. Only for 4.
The cell at the prophase.
The cell at the prophase has 6 chromosomes. These are only the conditions given. Based on the conditions, you are drawing the diagram. You are getting 2 marks only for drawing the diagram.
All 4 combined.
If all the 4 conditions are correct. Understood? 4 lines, leave 4 lines, 4 to 5 lines for the diagram. The diagram should not be that big. Number 3, number 4.
Every single shows a different stage, so we have to draw 4 different diagrams.
9. You are drawing only one diagram. You are drawing only one diagram based on all the 4 conditions. You have to think of all the 4 conditions and draw one diagram. Number 4. 1, 2, 3, 4. Number 4. What are homologous chromosomes? What are homologous chromosomes? What are homologous chromosomes? Name the longest phase of, same question, all I'm giving 2 marks, so every question will have 2 marks. Name the longest phase of cell cycle. Name the longest phase of cell cycle. That is 4.
Cell cycle.
Cell cycle. 4, 3 lines for the answer, go to 5. Go to 5. Write two distinctive difference between mitosis and meiosis only based on these 2 features you will write. Between mitosis and meiosis based on point number A, daughter cells. Point number A, daughter cells. Point number 2, need. Need or importance. Need you write. Why mitosis? Why
Can you spell it out?
N-E-E-D. Need. Daughter cells and need or importance. That is number 6 it is?
Yes, 56.
Number
5th.
5th. Okay, write this 5. So 2 marks each, 2 marks each, so it is 10. Don't take too long. 5 to 7 minutes, 10 minutes should be more than enough. Write fast. Diagram drawing only with pencil.
Nucleus.
Key terms always to remember that you write. Make your answers very specific, writing numbers and points wherever necessary. Highlight the key terms if there. Don't write in big long paragraphs. Science, always avoid writing in big long paragraphs.
To genetics.
You all only said, I wanted to start photosynthesis, but you all said genetics. एक ही डायग्राम ड्रॉ करना है.
Do you also mention the name
of
the stage?
It's mentioned in the question.
Oh right, last. Sorry.
No, the stage is not mentioned. The stage you are supposed to identify. One stage is mentioned which gives you an idea about the chromosome number. Done?
Almost.
So
My pencil
you
broke.
are supposed to have.
My pencil broke.
So पूछो किसी से पूछो.
मेरे पास एक ही है.
कोई नहीं मिलेगा।
Make it fast. Jaldi, jaldi. You should know the concept. When a very simple elementary questions there है, you should be able to write fast. Okay? Over. All done.
One question.
हम नेवी भी कर रहे हैं नेवी
क्या नहीं हाँ diagram whenever you are drawing to label basic 2-3 parts label करना ही है एक नहीं आ रहा Don't take so long. I'll give you last 5 minutes more. Chalo. 5 minutes. In 5 minutes, all of you should be over. All of you have to show me your answers now. Done? Yeah, don't tell me. Don't ask me anything.
सेंट्रल जोन को आया था मुझे सेंट्रियोल याद है मुझे सेंट्रोमियर याद जो क्रोमोसोम है सेंट्रल जोन को याद
नाइन्थ में था
होगा वाकई चला गया तुमने
हम होमोलोगस सिलेबस में था
हाँ
आई थिंक दैट इज इन जीन चैप्टर जेनेटिक्स
आई मीन होमोलोगस
जाके मोबाइल लेके अच्छा है क्यों
मेरा फोन
You have your phone silent पे है done
हम 2 मिनट
we also mentioned this stage diagram that you are drawing diagram में only as per the 5 conditions see I have given you this form another format of a question is draw the diagram for such and such stage and label such and such parts they will give you that type of a format also and they will give you this nowadays in the last 2-3 years new formats of questions are coming up so get prepared with the concept very clearly. So in this type of a question, what happens? You have to read the question, get the idea what should the stage be, plant or animal cells, and how many number of chromosomes I should take. How do I show the placing of the chromosome? So what are the prominent parts? Definitely the chromosome, centromere, etc. So you have to label.
रेजर किसके पास है
And complete learning.
तो रख ले भाई
बढ़िया होना चाहिए. Those who are done, please show. Those who are done, show fast. Bharat, how many more left? 2 more. Priya, 1 more. Divanshi, how many more?
Ma'am, I was absent for the sales chapter and I did revise the chapter.
The entire chapter you were absent?
Yes, no, the last part.
Acha.
I did revise, but I didn't recall much.
Aditya, done? You are taking too long. Chalo now, time up. If you don't remember certain things, you will not be able to remember right now. That means you need to read. Show your answers. Thank you. Need I had asked you. Why the process? Then
For now.
improve. Transpiration. Okay. It is correct. But you may not get full marks at this time of time. What is it? लिखिए
What are second part? What are the second part? What is my time?
Dance to know.
What is influence?
In second one, I don't remember. Diagram is very clumsy. Correct है, so then इसके लिए only one. Discuss करके अगर टेस्ट बना हुआ appear which will be subjected and analyzed on a basis specific issue. You can understand from this. Study this question please.
जेनेटिक्स में कुछ तो मैथ्स का भी है क्या? एक्स और वाई का
चलो पास
I'm just wondering if this type of words. The wrapping around of the DNA around the specific word. See, when I see my friend,
I can tell her hi. So when I meet you, can I say hi, hello? There's a format for everything. When you are expressing, you can use any word. But when you are writing, there has to be a specific format how you write on the answer. नाम क्या है? Why this process should take place in all time? ये process नहीं होगा तो ये नहीं बनेगा.
सच्ची हमारा बाइक फॉलो
What is this? The concept तो clear होना चाहिए ना. नहीं क्या है? What is writing? इसके साथ इसका क्या connection? Understanding, but the answers will come retention बढ़ा. You should know what they are thinking. Part of the question
हमारी माँ बोल रही थी कि जो आप किस दिवस में लिए मुझे आप पढ़ो.
What are the proteins present in the centromere? इसके साथ एक और question मैंने किया था. What are the proteins present in the centromere के साथ? What is its function?
Yeah.
सेंट्रोसोम अरे वो एक्शन भी है
Histone protein है.
नहीं यार
नहीं. क्रोमोसोम है कहाँ?
स्पिंडल है ना
Histone है.
क्रोमोसोम कहाँ?
मेरा किस स्टेप होगा?
ये क्रोमोसोम तो पोल्स कहाँ है? सेल के पोल्स कहाँ है?
आई काइट जैक्सन
देखो आप क्या बोला है?
मेरे दोस्त की डिस्कस एंटायर नाउ मेरे दो बार
सेकंड टॉप ऑर्डर
नहीं
इसके बाद बहुत ही दो साइड और एसटी से लेकर
मैं ऑनेस्ट बात मैं बता बता नहीं है ना सही क्या है भूल गया इसलिए नहीं पता लिखो
तो वही समझ रहा
बस वही था कैसे
वो किया
बस वही
कोई
understand what is need.
अरे नीड
I said need.
I gave you the spelling also.
I thought you said need.
वो पहले
I'm so sorry need क्या है?
12th कंप्लीट करो उसके बाद नीड का सोचो. अभी पहले तो नीड सीखो.
ये कर रहा है अभी.
यूरोस्पेस
नहीं आता है.
चेंज का मतलब पता नहीं
So math discussion.
संडे को भी कोई और वजह से हो गया अगर ये लोग कहीं से
क्या करते हो
इसको बुलाया तो ऐसे तीन से कर
देखा सारे सब करेक्ट है.
मेरा तो ऊपर किधर ही है
करेक्ट नहीं
हम मुझे प्लान
सोच तो पक्का करें
हो जाएगा लुटेगा
हम लोग कर रहा है वीडियो देख रहा है बोले तो कितने नंबर में लेकर आया
सोच तो पक्का करें
कर रहा था मुझे बता रहा था सब करेगा
तुम बता भी रहे हो तो टीचर.
अबे क्या बता
वो बोले इस साल में देखना न्यूज़ में आ रहा हूँ ना कोई स्टेशन के
What are
और सुनने बेल करेगा
nucleosome क्या है?
बे हमें समझे पूरे क्लास में
नहीं
तो भी nucleosome
बंदा पढ़ा ही नहीं सबसे फील करवाते हैं
अभी बाकी अभी जो holidays आएगा, let me tell you, ask your parents not to plan for any holidays. This is their plan, but you have to be serious. And for the rest, all of you.
I will do
My
too late
I went for all.
मैंने एक दिन
इसके पहले
मम्मी मैं तो
करवा दिया
सब बन क्या?
मैं तो साइलेंट हो गई
क्या टेंशन ले रहे हो?
अरे छुट्टी में
पहले तो टेंशन मत ड्रॉप करो कभी भी.
बिहार
पेंसिल दिया खुद भी पेंसिल रख
बिहार
नहीं
चेन्नई
हाँ भाई चेन्नई छुट्टी मनाने के लिए चेन्नई गई। एक घंटा
भाई अभी फ्राई बेस्ट है केरला क्या पढ़ने के लिए
नहीं देखने का
टू माइट आफ्टर
थाईलैंड में भी आईसीएसई का देखो पता है हाँ सिलेबस है ना वो सिलेबस वो कुछ भी नहीं चाहिए
देखा था मैंने
All of you will look for the answers. जो भी नहीं लिखा, जिन लोगों ने भी answers fill in नहीं किया, go back, look into the text, look into the notes, and fill in all the answers. Fill in all the answers. My God.
वो अच्छा नहीं है
नंबर
I think it is very good looking है क्यों बहुत गंदा डायग्राम है वो क्या बोल रहा हूँ बहुत सुंदर होता है ना ऐसे अच्छा नहीं बना पाऊंगा
Abhipray, ये diagram is not at all appropriate. What is the diagram? It has to be spindle shape.
What is that?
अच्छा, ये फाइव भी है, spindle shape भी है. How, where you are going to go and justify?
डायग्राम बनाते हैं
The diagram that you have to draw has to be spindle shape with a slight file. Diagram इससे ज़्यादा बड़ा छोड़ना ही नहीं है.
बच्चे देखो मैं भेजा
Diagram इससे ज़्यादा बड़ा होगा ही नहीं.
बड़ा
बड़े डायग्राम
होगा. And then how many chromosomes I told you? 6 chromosomes in the prophase. So 6 chromosomes. Quite a number of yeah. All of you will check into those questions, similar questions. Paper में Sunday को जब आएगा. 6 chromosomes. When I say all of you have made only she did it correct.
I did it
also did it correct. She also did it.
I also did it correct. थोड़ा
हमने करेक्ट किया पर विद स्पेक्स also are not able to see correct.
हम एक मार्क
So नहीं, this is ये कोई bargaining थोड़ी है कि एक दे दो दो दे दो. Now see 6 chromosomes means what? It has to be 6 chromosomes I said in the prophase stage. Meaning हाँ, so it should be duplicated. Meaning यहाँ पे 6 होगा, यहाँ पे 6 होगा. So if this is and then where is the pole? If this is the pole, the chromosome has to be drawn this way. If this is the pole, the chromosome has to be drawn this way because the centromere will attach with the spindle. If you remember the stages that we had drawn in the class, the centromere will be pulled this way. The centromere will be pulled this way. So this is the diagram. That's why I
हमारे को यार मत चेक करो छाप कर
इसका
इसका
इसका खुद कर रहा हूँ मैं
That's why I did not give you any mark because I was not sure that whether you all will be able to answer. Now this इस तरह से हमेशा the centromere should be towards the pole. Centromere should be towards the pole when you draw the 6 chromosomes in the prophase. मतलब 6 duplicated 12 हो गया in the metaphase. So here 6, here 6.
है तो भाई ले
Fast, fast, come back fast.
क्या बॉटल यार तेरी हर बॉटल लीक कर दी है
भूल गया थोड़ा सा नहीं बोलना
Spindle fiber, this is.
Oh, I mentioned that also, and I've given answers.
अच्छा.
तो आंसर क्या है?
This is the protein. What is its function? Helping the function. Yes.
अरे मैं नहीं अरे यार तुमने
Always you should write consisting of.
Parental chromosome.
No, I'm sorry.
Paternal, maternal.
So
Both the parents is wrong.
Parents you should not mention. Specific words mention करना है. So it is a pair, no, like this. This is the maternal, this is the paternal.
One mark.
Next,
answer है formation of gametes. Formation of gametes. Differentiate में.
presence in the reproduction नहीं होगा तो कितना और इसका आंसर क्या है? I am not able to find it. Question to protein is present.
Central.
So I don't need
Mitosis का correct.
I am not giving you marks.
Centriole.
Centriole. In the class also you will have enough.
So I got
Walk. I'm not going to tell you prepare this, come and prepare that and come. This you
Why
have
not
to be on your own. हो गया.
ऐसी ना
I know. मेरे को bio में 75 आया था मैंने try किया.
नहीं लिखना. Straight away this word should be there in the answer. Okay. Drawing practice at times.
Method अच्छी हो.
वो तो मैं बोल
Why?
तुम दोनों से अच्छी हुई
You know.
तुमने भी बटरफ्लाई बताया पर तुमने बटरफ्लाई की तरह
लिखा कि नहीं लिखा?
लेकिन हम करेक्ट है
नहीं करेक्ट नहीं होता.
आप भी खराब
गवर्नमेंट को दिया ना.
हमने बटरफ्लाई को जाते हैं
अब तक
अब मैम हम लोगों को भी
What is this word?
वो मैंने सही करा
हाँ तो वो सही है.
तो मतलब हमको भी
ना मिला.
हमारा माइटोसिस का इंट्रो मुझे 2 मिले दो
कितना टोटल?
3 होता है ना 9
नहीं टोटल कितना है टेस्ट?
दो ओ शिट मुझे 2 नहीं मिले
On the first question I wrote is right.
फुल तो नहीं मिलने वाला है
Surrounding the district.
चलिए अच्छा लगता है
मीन में मार्क्स पूछो.
Proteins in the centrosome. What are
दो
the
था टोटल मार्क्स कितने
proteins in the centrosome? What is the protein in the centrosome to write, class? What is the protein in the centrosome to write? Centriole or microtubules. But microtubules I did not written in your book. So सही है. Microtubule दिया है. It is given. तो microtubule अगर दिया है तो centriole, microtubule, any of these answers you can write. But centriole तो लिखना है. Microtubule if you write is even more specific. Proteins. Let me discuss all the answers as I correct this book. What are nucleosomes? What are you supposed to write? Check your answer. What are nucleosomes? What is that? The packaging unit in which the DNA wraps around the histone proteins. The packaging unit in which DNA wraps around the histone proteins. That is known as nucleosome. What is meant by histone? Octamer. Histone protein that contains 8 subunits. Histone protein that contains 8 subunits. Histone octamer. What are proteins present in the centrosome? Centrosome is what? What is centrosome? You get a question in your paper. Differentiate centrosome and centriole. क्या लिखना है?
Centriole and the proteins present in
Centrosome is the cell organelle which helps in cell division. Centriole is the protein present in the centrosome which helps in spindle formation. So I asked you this, centriole to write down. They help in cell division, they help in spindle formation, specific to mention. The diagram would be a stage of anaphase to draw. The diagram that you draw, except for 2 or 3, the rest, the diagram that you have drawn, I am shocked and surprised. So the diagram that you draw, I think the best diagram that was drawn
Me.
was
the wrong था.
next. The diagram,
Me?
no, too long. That became a barrel almost. Devanshi ka diagram I would say much better than the rest. So लगभग इस तरह का diagram होगा, not too long, not too short, not too small. एक standard
It
size.
should be that wide.
So I'm saying comparatively, like it has to be little bit oblong, little bit or oval होना चाहिए था. But still for this diagram, teachers would give you out of 2, 1.5 मिलेगा. But for the rest, half 1 से ऊपर नहीं जाएगा. Then this is what are homologous chromosomes? Homologous chromosomes क्या लिखना है? A pair of chromosome consisting of the maternal and the paternal homologue. Pair of chromosome consisting of maternal and paternal homologue. Longest phase of the cell cycle. This more, I think all of you got it correct. G1 phase is the longest phase. Mitosis, meiosis का difference मैंने पूछा था. Basic difference I asked you. Mitosis, meiosis based on the number of the mitosis, meiosis based on the number of daughter cells. Very simple. 2 and 4. Mitosis 2, meiosis 4 cells. And significance. What is the need for mitosis? Repair, growth, maintenance of the body tissue cells. That is it. Replacement, maintenance, growth, etc. And what would be for formation of gametes, reproductive cells or gametes. So this is what has to be written. Okay. Ansh, did you write down wherever you did not write down the answer? Did you write?
Place the October next.
It's serious. See, when you are understanding a topic, that is absolutely different, and how you write on the paper, that is absolutely different. The board does not have any machine to scan your brains. Only what you write, based on that you get marks. Your answers have to be specific. Your answers have to be correct and right. Yes, Aditya, क्या doubt है पूछो?
He has written something in my book.
What? कौन?
मैं वो ऐसे क्रॉस किया मैं
किसने?
डायग्राम आपने उसके ऊपर बस ट्रेस किया.
क्या हुआ?
He has drawn something in my book.
किसने? अंश ने. And you very happily love to sit also with him always. हाँ? अंश क्या किया उसके book में?
हम ड्रॉ किया था क्रॉस.
हाँ.
उसकी ट्रेस किया हमने.
उसको ट्रेस करके फेसेस बनाए तुमने. हाँ? The next test that I give you, उसमें अगर सारे answers correct नहीं है, this type of mock test I will every time give you. 10th standard में हो, I'm not supposed to tell you that ये पढ़ के आओ बच्चे and वो पढ़ के आओ. You are supposed to keep reading, studying every time. I'm not giving you any homework. You are not small children that I give you 15 questions, go read के practice करके आओ. Nothing. Just read. Every particular chapter, how many times minimum you should read? Minimum 5 times पढ़ना ही है. If you want answers to be very fluently written in your answer paper every time, every particular chapter has to be read, just read and understand. I mean, you should understand and read, not just read and understand. Minimum 5 times. Now let us move on.
इस बार phase आना phase था.
डायग्राम
टेलोफेज
अगर होता तो ये स्पिंडल इतना नहीं दिखाओगे.
One cytoplasm
न्यूक्लियर मेम्ब्रेन आ जाएगा.
Can we do absorption by root division because it's only half an. Then next fresh start करेंगे. इसमें सभी चैप्टर में करते हैं जैसे संडे को टेस्ट करना है.
कल 3 घंटे.
No, let's start then.
हम आधा घंटा है वीकेंड स्टार्ट.
हम तो 3 घंटा है तो कल स्टार्ट करेंगे.
No, ma'am, let's stop.
Okay. I did not I give you any question on absorption?
No, ma'am.
No, you give.
Answers दिया भाई.
I will just take a short oral revision लेते हैं, but let us start with this. एक थोड़ा introduction करते हैं. Let us start. We'll talk about the details tomorrow. Not a problem. Absorption में what are the most important things that you should remember? Absorption. When you study the chapter, definitely you will get questions on a concept of endo and exoosmosis, hypertonicity, hypertonicity, hypotonicity. ये basic terms.
Hypotonicity, ना?
Hypotonicity, hypertonicity इसके concepts होने ही चाहिए. Raisins kept in the water swell up. What is the reason?
Hypotonic solution.
Raisins kept. I will just ask a round of questions. All of you just try to answer. That is a simple revision I want to take right now. बाकी chapter पढ़ो अपने आप पढ़ो. I'll start with Devanshi. Raisins kept in the water swell up. What is the process involved? Endoosmosis. Define the phenomena. So define the phenomena. Endoosmosis define नहीं करोगे. You will define osmosis. Define osmosis. Define osmosis. The movement of semi-permeable membrane. What is meant by a semi-permeable membrane?
Semi-permeable membrane is something that it's a selectively permeable membrane that only allows some particles to pass.
Only allows solvent.
It allows only solvent molecules, water molecules to pass through. It allows only solvent molecules or water molecules to pass through. Give 2 examples of semi-permeable membranes.
Parchment paper and bladder.
Animal bladder, both bladder, egg membranes, animal bladder. This is you can write down. So now, then next is what is the difference? Write one major difference between osmosis and diffusion.
The diffusion, osmosis happens to a semi-permeable membrane, but for diffusion, there needs to be contact.
There has to be
There needs to be contact
between the molecules. For osmosis, the movement is always through a semi-permeable membrane. For diffusion, the movement or the molecules showing diffusion always are in contact. There is no barrier, no molecule. Examples you can always remember. Now, for absorption by roots, the concept of those stem and those stem cuttings and all those things you have to remember. Now, for example, for absorption by roots, what is turgidity? What is turgidity? Define turgidity. What would you write? Define turgidity. The condition of a cell cannot accommodate any more water. That is called turgidity. When will a cell become turgid? When a cell is
when the process of hypotonicity.
When is a cell turgid?
When the cell is completely filled with water.
What should be the surrounding of a cell that it is turgid? The cell will be in a hypotonic solution. Define hypotonic solution.
The solution which has more concentration of solute, more concentration of solute than the cell present.
More concentration of solute meaning
solvent.
More concentration of solvent as compared to the cell. Clear hai? What is root pressure? Aditya, will you be able to answer? What is root pressure? Define root pressure.
26. Homologous chromosome, they are only written parent in definition.
Parental homologs लिखना जरूरी है.
मैंने लिखा था.
कहाँ पे? Maternal homologs लिखना जरूरी है.
Maternal नहीं दिया है.
तो क्या दिया? ये definition क्या
Pair
है?
of corresponding chromosomes of the same shape and size obtained from each parent.
हाँ, तो obtained from each parent जो हम लिखते हैं, that is common. Try writing the maternal and the paternal homologue of the same size, arrangement of the chromosomes. हाँ, क्या पूछा मैंने अजित्य? हाँ, define root pressure.
Root pressure is the pressure which is formed in the root tissue due to cell-to-cell diffusion.
And this helps in ascent of sap. What is root pressure? The pressure which is developed at the root cells due to cell-to-cell osmosis, and this helps in ascent of sap. How can root pressure be measured? You may get a diagram-based question about that manometer, the mercury and the manometer. Remember? So the stem is there, the stem is cut, and then the glass tube is placed, the glass tube bent, glass tube mercury recover is made. So these type of questions. What is the difference between root pressure and transpirational pull? Transpirational pull कैसे define करोगे? Transpirational pull will be the suction force developed in the xylem tubes. Today also मैंने वो explain किया था. रुको, I'll come to it. Transpirational pull is the suction force developed in the xylem tubes due to continuous transpiration, which helps in absorption of water from the soil by the root cells. So this is what to write. Next is define tonicity.
Tonicity is the concentration of solute, relative concentration of solute in the surrounding water of the cell.
What is tonicity? Tonicity is the relative concentration of, therefore define
Solution.
relative concentration of solutions. You can also
That
write.
determines the direction and extent of diffusion is called tonicity.
So what is tonicity? It is a relative concentration
That determines the direction and
so
extent
you
of.
have to understand relative concentration मतलब what? Semi-permeable membrane के दोनों तरफ concentration क्या है? That is the relative concentration. Keeping in mind the semi-permeable membrane in between. It is the relative concentration of two different solutions which will determine the movement of the water molecule. So water molecule अगर इस तरफ जा रहा है, so definitely this is hypo and this is hyper. वो point clear concept होनी चाहिए. What is meant by tonicity? Define turgor pressure and wall pressure. Turgor and wall pressure. I have shown you so many figures.
Turgor pressure is the pressure which is
Exerted
towards the walls from the inside.
Exerted by cell contents.
Exerted by cell content, while wall pressure is the
Exerted by the cell contents on the cell wall.
on cell.
Answer complete होनी चाहिए. What is turgor pressure? Pressure of the cell contents on the cell wall. And what is wall pressure?
Wall pressure is the pressure of the wall towards the cell contents of
Pressure
cell.
of the cell wall.
Pressure of the cell wall towards the cell contents.
Cell contents, towards the cell contents, right? Two uses of turgor pressure in a plant. Two uses of turgor pressure in a plant. Turgor pressure helps in so many different aspects of a plant. Number 1 and 2, tell me. Number 1, what did we study? Turgor pressure helps in what was that? Turgor pressure helps in opening up of the stomata, opening, closing of stomata. Turgor pressure help करता है. What else? If I talk about germination of seed, the breaking up of the seed coat.
Helping in the ground process.
The breaking up of the ground and the growing up of the roots. That is also turgor pressure. Stiffness of the plant parts. That is also turgor pressure. What is osmotic pressure?
Pressure developed by the
Osmotic pressure. The moment the definition लिखना है, क्या कौन सा experimental setup याद आता है?
दो बीच में separation और दो bubbles type.
No. Osmotic pressure में कौन सा setup याद आना चाहिए? The thistle funnel है, thistle funnel के ऊपर वो piston है. So thistle funnel के बाहर भी water है, but then the water is not moving inside the thistle funnel. So वो setup immediately याद आना चाहिए. Then so book के pages you have to go through a number of times and only you'll be able to understand. Tell me the pressure which is responsible for opening up and the closing of this one mimosa. Which pressure is responsible for that? Folding of the leaves of mimosa में which pressure majorly plays an important role? Turgor pressure. Okay, now Bharat, I just now told about a concept of absorption. No? Absorption, hypotonic, hypertonic. Can you define hypertonic solution?
Hypertonic solution is when the
Solution.
solution has higher concentration
Of
solute than other atmosphere.
then the cell.
Than this.
Then the cell. Okay. So when a cell is placed in a hypertonic solution, what will happen to that cell?
It will experience high concentration.
A cell is placed in a hypertonic solution. What will happen to that cell?
It will get higher pressure.
क्या हो रहा है?
What?
A cell is placed in a hypertonic solution. I think you can put it up from there also. Wait, sit there. He's getting the remote.
This process of exosmosis.
A process of exosmosis will happen. A process known as exosmosis will happen. So now that the process of exosmosis happens, if the cell is still kept in that hypertonic solution for another 6 hours, what may happen to this cell? Yeah, the 27. What will happen to that cell? Okay, next. Aryan, no? The cell is placed in a hypertonic solution. What will happen to this cell? Cell is placed in a hypertonic solution. It will become flaccid. Why? Which process is happening? Exosmosis. The cell is still kept in this hypertonic solution for another half a day. What now next? Plasmolysis. Define plasmolysis.
It is the condition in which there is no water in the cell and that cell membrane is detached from the cell wall.
And the cytoplasm is shrunken. This condition is known as plasmolysis. What is the entire pathway? Can you name the group of cells, entire pathway through which the cell moves from the water to reach the xylem? पहला क्या है? The first is
to the
root hair cell.
Then to the epithelial cell.
Epidermal. हाँ. Epidermal के बाद
Epidermis, then endodermis. There's one thing left.
No.
Then endodermis.
Cortex.
Epidermis.
बोला ना,
रुको.
Epidermis,
Epidermal के बाद cortex. Cortex के बाद
endodermis, pericycle, phloem, xylem.
नहीं, phloem बाद में.
only xylem.
Phloem पहले and then
Xylem.
xylem. So this cycle, this entire arrangement is very important when you are reading this chapter, because the placement of the cells, arrange them in a logical sequence. This question is very common. Arrange cells in the arrangement of the cells in a logical sequence के लिए this answer is very. What is osmotic pressure? He could not define. This is very important. Osmotic pressure. What is osmotic pressure?
Osmotic pressure is the minimum pressure that must be applied.
That prevents.
That must be applied on the solution that prevents the passage of pure solvent into the solution when the two are separated by a same permeable membrane.
Okay. So now that we are talking about osmotic pressure.
There's one more definition. There's a measure of tendency of osmosis.
No, the previous definition you should always write.
That
What is osmotic pressure? The minimum pressure exerted that prevents the movement of the solvent into a solution. The two are separated by a semi-permeable membrane. ये definition लिखना चाहिए, which is the most accepted definition. These are the acceptable. And then when we are talking about the different type of setup for the concept of capillarity, in which type of glands in a capillary force? याद है? What was capillary force? Capillarity. What is capillarity?
I don't remember the definition, but
अच्छा, basic बताओ. What is capillarity?
Stem or a tubus or anything, even
No, with reference to xylem बताओ capillarity.
When a xylem, the stem is put inside inside, let's say, water.
The tendency in which water enters into the xylem tube. without any extra pressure. We are not talking about transpiration or full root pressure, kuch nahi, small amount of water that enters. So the
Slightly
capillary.
above the normal level of the beaker.
Sorry. So now the capillarity will be more in the xylem tubes of a shrub or in a tall tree. In a
Tall
tall
tree,
tree,
because the xylem vessels are much
narrower.
smaller.
Narrower. They are
The radius is smaller.
narrower. So when we are talking, what is imbibition?
Imbibition is the phenomenon by which living and dead plants absorb water by surface attraction.
Very good. Next is the entire concept of to study the processes of absorption by roots. What are the three major features of the root cells? What are the three major features of the roots which are
large surface area of the roots due to the presence of root hairs, a higher concentration of the cell sap when compared to the outside hypertonic medium, which supports the process of osmosis, and then it was the cell wall being a hydrophilic substance attracts water, and the presence of cell wall attaches water to it. So that
next, simply you should write it. That is a question. Simply you should write the cell wall and the cell membrane features, whether it is semi-permeable है कि freely permeable.
So ये cell wall and
है.
semi-permeable is cell membrane.
Okay. So 3 mark के लिए just basic writing. No need of so much explanation. जरूरत ही नहीं है. Next is when we are talking about the concept of root pressure, name the two prominent pressures which are helping in the ascent of sap.
Transpirational pull and root pressure.
And root pressure. Transpirational pull and root pressure. What is the difference between cohesion and adhesion?
Cohesion is the attraction between molecules of the same kind. Adhesion is the attraction of molecules of two different substances. So the force of attraction.
Explain cohesion and adhesion in the xylem tube.
Yes, ma'am, that is what I was doing. So the attraction of water molecules to each other is known as cohesion, due to which they move in long chains together, and the attraction of the water particles to the wall of the xylem is known as adhesion. Which helps in the process
the
of
water molecules always move in a
straight line.
In a column. They always move in a column. So this many concepts that I have asked you right now, all these things definitely you should know, and all these experiments. One experiment from this even in your boards is for sure. इस पेज में जितने एक्सपेरिमेंट्स है इसमें से एक तो 5 मार्क के लिए आएगा ही. Okay, so ये whatever I had discussed, this many concepts, you have to read the chapter quite a number of times till you appear for the pre-boards. So at least 7-8 बार तो पढ़ना ही चाहिए. That time management you have to do. You are a 10th standard child. Time management enough you have to learn now. Okay. So let us start with this chapter. We don't have too much time, but I want to start with this chapter today. So we are talking about the concept of genetics, fundamentals of genetics that we study. So before we enter into the concept of what are chromosomes, what are genes, before that, what is genetics, what are we trying to study? The entire study of the genes and the characteristics. The word genes not new for you. You already know that. What is the meaning of the word genes? Where is it located? Which part of the cell, chromosome, etc. we are talking about? So this two definition, first of all, to understand, when we are talking about one specific definition, that is heredity. What is heredity? This definition you are asked in your exam. What is heredity? What is the definition of this word? Always you should know the process of transmission of characteristics from parents to the next generation. So here we are talking about two words, that is parents to the offsprings. Parents to the offsprings. Offsprings always mean the next generation organisms that we are talking about. The transmission of characteristics by the way of chromosomes, we are going to understand. So what is the meaning of heredity? Hereditary characteristics, when we say, what does that mean? The characteristics that we inherit from our parents. So transmission of characteristics from the parents to the offsprings, that is what is known as heredity. But to study the concept of genetics, what is the first thing that you should know? You have to remember the concept of chromosomes and the DNA. So to talk about the concept of chromosomes and DNA, first of all, again, I think of a cell, the concept of cell division and all these things we have already studied. So I now talk about a cell. The cell has got the nucleus, and the nucleus has got the thread-like structures. And now to talk about the thread-like structures, how are these thread-like structures? I may talk about that these thread-like structures when they are condensed. What did we learn in that chapter on chromosome, structure of chromosome? How did we define the chromatin and the chromosome? What was the basic difference between the chromatin threads and the chromosome?
Chromatin threads are condensed form.
Chromosomes are highly condensed form.
Chromatin
Coil and
and
chromatin threads. And chromatin threads, how will you define? Chromatin fibers, how will you define?
condensed form. When they are
Chromatin fibers, how will you define? The extensively long uncoiled thread-like structures. They are extensively long. That's the reason they cannot be easily seen under the microscope, the chromatin threads. But the same chromatin threads, when they are extensively coiled and condensed, now they look shorter, now they look broader, and this is what we are talking about as chromosomes. So if I say that the chromatin and the chromosome, they are almost the same, then should their composition also be the same? Yes, their composition should be the same. We are not talking about absolutely two different things. I'm saying it is the chromatin only which was there in a new cell. It is the chromatin only. So if I talk about this, maybe I may talk about that how did this cell develop such type of structures. Maybe this cell when it was newly formed or young, what type of a stage of the cell I may define this as. This may be considered as a stage known as telophase. In the telophase, what is happening? You've already come across the uncoiling of the chromosomes happen. And the chromosomes now start uncoiling, resulting in the formation of extensively long, thin coiled structures. So this can be termed as chromatin threads or chromatin fibers. Okay. So chromatin threads, chromatin fibers present where? In what type of a cell? A cell which is newly formed after the division. And what about this cell? This is mature cell. This is a mature cell which is now ready for the process of division. This is a mature cell ready for the process of division. Okay, we come to this concept of chromosomes. Then what are chromosomes? Remember the definition. Some of these basic words and definitions in different chapters that you come across, concept wahi same rahega. So I'm talking about chromosomes. What are chromosomes? Extensively coiled chromatin threads. Now, looking into a small portion of the chromosome, try to understand what is this. Chromosome made up of, this chromosome is made up of a highly coiled helical double-stranded structure. So what is this highly coiled helical double-stranded structure known as? This highly coiled helical double-stranded structure is known as DNA. And what is the full form of DNA? I don't think I have mentioned now. Deoxyribonucleic acid. What is the chemical nature of this molecule? This is a macromolecule. In the previous chapter we had studied. This is a macromolecule. What is its chemical nature? Acidic. How do you know it is acidic? Because it has got a great affinity for the histone molecules. And the histone molecules are, no, you cannot write it is in the name. This is not the answer. The answer is, since it has got a great affinity for the histone molecules, which are basic. Nucleosome, I gave you a question right now. What is nucleosome? The DNA wrapping around the histones. So, here the coiling I told you just now. Coiling is how the coiling, the DNA constantly wrapping around, the chromatin constantly wrapping around the histone, and that is how we are talking about the
histones are bases.
Histones are proteins which are basic. Histones are proteins which are basic proteins. positively charged. So now that we are talking about basic proteins and the DNA is acidic, and that's the reason the affinity. That's the reason the binding. So this DNA molecule that I talk about, what is the full form? All of you should again take a note. The full form is deoxyribonucleic acid.
Do they react with each other?
No, they don't react. They are stable compounds. They are not unstable. They are stable compounds. But since the charges are absolutely different, the DNA is made up of chemical substances, the overall charge is negative. And if you talk about the histone proteins, the optima, they are made up of positively charged amino acids more. They also have negatively charged, but overall charge is positive. So that's the reason the attraction. All over the DNA, the concept of nucleotides, all of you should be very thorough with. Now that I'm not talking about the concept of nucleotides right now again, what is this DNA made up of? If you get a question, write down the chemical composition of chromosomes. What are you supposed to write? Chemical composition of chromosome is DNA. What is the chemical component of the chromosome? DNA.
Sugar, phosphate, nitrogen.
No. Chemical component of nucleotide, that is pentose and etc. etc. What is the chemical composition of chromosome?
DNA and
Okay.
histone.
DNA and histone. So now that we are talking about the DNA molecule, which is in the form of a ribbon-like structure, which is double-stranded. Now this,
Wait.
what happened?
Mom, he said, "Huh," and everyone was scared.
And now that I'm talking about DNA, and ये तुमको सुनाई नहीं देगा.
Mom, no. He said, "Don't."
So now, when we are talking about the entire DNA molecule, the flat ribbon-like structure, helical, double-stranded, these are all the features. So, looking into the entire DNA molecule, I would say that every single segment of the DNA molecule is responsible for specific type of a characteristic that we show, and this characteristic can be morphological, can be physiological, can be behavioral. So what is these small segments of the DNA known as? And these small segments of the DNA, I will say that maybe one of these segment is regulating our height, one of these segment is regulating the type of certain type of an enzyme, another type of a segment is regulating the eye color, etc. So when we are talking about all through the length of the DNA, all through the length of the DNA will have such type of segments, numerous in thousands. So what are these segments known as? Now that I talk about this particular segment, what are these segments known as? These segments are known as genes. And how do we define the word gene? What is a gene? These are the specific sequences or the functional segments of the DNA. The functional segments of the DNA that encodes a protein, and regulates, a specific characteristic that we show, regulates a specific characteristic. Now, what is the meaning of regulates a specific characteristic? Say, suppose how much of the hemoglobin to be present in our RBCs, that is also because of the typical type of genes that we show, the typical type of genes that we have. So when we are talking about what are genes, all of you will take a note. All of you take out your notebook. Certain things you will write down, keep on writing down in your notebook. You are not able to write down proper answers. Take out your notebook. Whatever I asked you to underline in your book, you will underline, mark, write. Unless and until you write good answers, you will not get good marks. So what is the specific definition? Write down the name of the chapter and we talk about what are genes. Genes are the specific functional segments of the DNA, and these specific functional segments of the DNA are always known to carry information for a protein. That's why we write the word encodes a protein.
Where is the definition?
The definition will be here only. The definition of the gene will be here. कहाँ पे है? See there.
This definition.
Page number 26. This is page number 26.
Also given in the first one.
I don't go by the pages. I will go only by the topic. You keep on searching the topics there in your book. That is your work, not mine. Now, when Mila, है ना पेज 6 में 7 में है
जीन्स का डेफिनेशन
this boy is
बोर्ड पे भी लिखा है
हाँ
बोर्ड पे भी लिखा है
हाँ नहीं उसको और क्लैरिफिकेशन
Page 12.
page 20 I don't know it is page 26
12
in my
में दिया
book.
ना first
हाँ.
chapter उसमें
So definition of gene is here also in this book also it is there in this chapter also it is there निकालो gene genome is given together. So what is gene that you have to understand? Genes these are functional small segments of the DNA. Every segment of the DNA contains an information to form a protein. I may understand protein for hemoglobin. How much hemoglobin to be produced? Now I say, what type of characteristic this gene regulates? 3 types I just now mentioned. The genes will be regulating the shape of our nose, fingers, height. This will be morphological features. The genes will also regulate how much of the pepsin, how much of the hormone, how much of the hemoglobin, functional characteristic. It is because of these genes only I don't like orange and I love purple and I love to read and I don't love basketball. It is because of my genes.
Genes?
Yes.
So if a parent loves to play basketball,
That's the reason. You also will have to find out that if you love to read, maybe one of your parent does or your previous generation does, because it is flowing 7 generations. So when we are talking about the carriers, we are talking about that these genes that we inherit, it is not to understand that genes are not only regulating our eyes, nose and height. Genes are regulating every specific characteristic. The type of behavior that we exhibit, remember that is also because of our genes and that's the reason we cannot change our behavior. Now if I am rude, I can only change little bit. If I am too tolerant, I am too tolerant. I cannot suddenly one day become over smart. So why we cannot change behavior? Does it have an impact on our genes? Yes, only 5 to 10%. So environmental factors does bring about a change, but not too much. That's the reason. So always remember that these concept of genes that we are talking about, what is the basic definition? All of you should write this.
Yes.
Functional segments of the DNA that encodes a protein. And these proteins will be responsible for what characteristic we exhibit. And I just now told you characteristics that we exhibit are of 3 types. Overall, every characteristic that we exhibit. But you will also find that the characteristics that you exhibit also matches with your parents' why? Heredity. Transmission of characteristics from parents to offspring. We will continue with this chapter tomorrow again.
तो क्या लिखा है भाई
definition.
हाँ पर क्या लिखा अभी प्रति को कितना है इतना ही तुम्हारे भी कुछ बायो की से कुछ नहीं होता 79 है सही बोल रहा से कुछ नहीं होता 79 में आने
उसको कितने में से लेके बोला
79 नहीं पर उसने बोला कि नोट से कुछ नहीं
मैं नहीं करने वाला देखो
वही तो एग्जाम में नोट्स लिखने वाला है 80 में से अगर तो नोट्स में गलत लिख दिया तो
इसको भी 99
99
out of 80 में से out of 100
92 है बोला
इस में
72 मिला इस बार ऐसे
क्यों
इस बार मिलता है ना प्रोजेक्ट
मुझे मिले कुछ तो
ज्योग्राफी में आ गई
कितना
94 अच्छा
ये तो ये सारे बने
मेरा हाईएस्ट ज्योग्राफी था जाओ
उनसे कुछ नहीं आता उनसे कुछ भी नहीं पढ़ा
मुझे छोड़ के
थैंक्स
देख पेन छोड़ के चला गया
कौन कौन
भाई कितना लिखा है टेबल पे यार
ये